Okay, good morning. Thank you for the organizer to invite me today to present an update on Eyevensys technology. So quickly, we are a non-viral gene therapy platform, and our goal is to develop treatment for ocular disease. So we are a French company, as you can hear from my accent, but we're also incorporated in the United States, and we are in fundraising mode, of course, because we want to accelerate some of our assets for wet AMD and dry AMD if possible. And we're well funded so far by mainly Boehringer and BPI France, but again, we're in fundraising. So the technology, as you remember, it's based on plasmid DNA, which is encoded to produce therapeutic protein. This plasmid DNA are injected through an ocular device in the ciliary muscles. The ocular device is linked to an electrical pulse generator, and the strategy is to do electrotransfection, and for us, we did not change the name, it's still electrotransfection, and to produce therapeutic protein directly in the vitreous and in the choroid. So the big accomplishment since we are presented at OIS was the device. We have definitely improved the device design. This is because we went in the clinic in non-infection uveitis to demonstrate safety of electrotransfection, safety of the plasmid, but also collected information on the design. I'm going to try to get that video work. So it's just a type of cartoon. We inject in the ciliary muscle, there's a little guiding tools there. We do electrotransfection linked to the ciliary, to the pulse generator, et voila, that's done. So the objective here again is to produce therapeutic protein directly in the vitreous and in the choroid, and to have durability at least for 6 months. Today, you know the problem with therapeutic approach we have today. This audience knows what we talk about. So what are we offering here? We're offering an innovative approach. Our first indication we're targeting are wet AMD and dry AMD. However, we went in the clinic in non-infection uveitis because we had to demonstrate safety first and collect information on the device. It's a growing platform. We can think about any type of protein. We can encode a plasmid to produce dual proteins also, and I will explain that in a minute. We can think about glaucoma and neuroprotection. And from a market consideration, think about that's a new administration, so we would get a new J-code, obviously, a new reimbursement. This is just quickly on the pipeline. Again, the non-infection uveitis program was there mainly to demonstrate the safety and the feasibility of the platform, but we are accelerating the anti-VEGF angiogenetic program. Uveitis, just a few words. We did enroll 18 patients, late-stage disease. The goal, again, safety. We did it. Few patients, we show some efficacy. I mean, I don't like to say efficacy, but signal of efficacy or biological effect. Again, that was a safety study, and to collect information on the device. The wet AMD program, we've built a plasmid to produce 2 therapeutic proteins, an anti-VEGF with aflibercept and decorin, which is a native protein. Here again, we know that decorin had anti-fibrosis characteristic as well as anti-angiogenetic characteristic. The goal here is to be able to produce 2 proteins at the same time, durability for 6 months, 1 injection. We have a few preclinical models, of course, we have done to show that we have similar production of aflibercept as if we would have with Eylea, but also with decorin, we show the anti-fibrotic effect. The GA program is a little bit different. It's a protein, also a native protein that we're producing. It's called transferrin. It's an iron chelator with antioxidant and neuroprotective characteristic. The idea is to protect the cells from dying. So once again, we would have to treat the patient early on the disease and try to slow down progression of the disease with an injection every 6 months or more. If you think about GA, you also think about RP and also retinitis pigmentosa, and also about glaucoma, protection of ganglion cells. And also, we have numerous animal models where we demonstrated efficacy and protection of cells, and also protection of the functions. So what have we done so far? We have done multiple animal models showing that we can express multiple protein, dual protein, sustained protection in rabbit for 6 months at least. We have shown safety in a clinical trial with 18 patients in late-stage disease uveitis. From a regulatory point of view, obviously we're a combo. We met with the FDA twice, so it's going to be a BLA filing. And from an IP protection, we are protection at least until 2037. So if you think about Eyevensys, think about new platform, disruptive, non-viral, gene therapy, non-virus. So it's a combination, again, of non-virus, but platform, and also electrotransfection, new J-code. So that's it. Thank you.